about today is some of the work that we've been doing in this area, which is really about how cancer interacts both with nerves and with chemotherapy. And so today I'll talk generally about degeneration, neurodegeneration, and how there are different types of neurodegeneration that we can distinguish. I'm going to then focus on one particular type of neurodegeneration, which is chemotherapy-induced neuropathy. And I'll talk about what that is and how it works. I'll talk about the basic biology behind that and how we're using understanding of the basic biology to try to develop new therapies that will help patients. And so that will be what we'll talk about today. So you've all heard about neurodegeneration, whether it's Alzheimer's disease, Huntington's, Parkinson's, and the picture on the left, the picture here, is really sort of what you most think of with that, which is the profound atrophy and loss of neuronal tissue that occurs. You can see here that this is this area of the striatum, which is completely gone in the brain of this person with Huntington's. Now, in that case, what's happened is cells, neurons in the brain have died. But there are other disorders where it's not the cells that die, but rather the connections between cells, the axons. And this shows a picture of the skin and these nerves that end in the skin. And you can count those nerves and see how much innervation there is of the skin, which allows you to feel touch, and how that is gone, even without a loss of actual cells. So when we think about loss of cells, there are a lot of different ways we can quantify this so that we can really study it in a very rigorous fashion. Most of these are imaging-based methods that can be made quantitative by an understanding of the process of how an individual cell goes from being alive to being dead. And so this is the timing at which these occur, some of the things we can look at that include loss of the membrane integrity, activation of some of the proteases that are going to be cleaving the cell and chewing it up, changes in the nucleus, it's fragmenting of the DNA, and finally, the final pathways in death. And this just shows some of those examples of what it looks like. And so we can readily count and study this in a very rigorous quantitative fashion so we can look at what's happening and what can be done to prevent these kinds of degenerative diseases. Now, when we think about neurons, the cell body itself is actually a really small part of it. So this, if you think about a cell, a typical cell, say a fibroblast in the skin, or a cell in your liver, or a blood cell, well, blood cells would be even smaller than this. There's the nucleus, and then the distance between the nucleus and this membrane is like 10 microns, and it's a really small thing. Now, individual neurons in your body have axons that can be as long as a meter in length, right? So neurons that are in your low back and innervate your toes, they have the cell body in your low back. They extend one axon down to your toe, and some of them extend a second axon that goes up to your neck. So that means these are very, very long cells, and that's an individual cell that's like 6 feet long, just as a little, did you know, giraffe neck axons are 15 feet, and blue whales are like 25 feet. So because they're so long, they constitute about, can be often 90% of the neuronal volume. So for neurons, unlike many other cell types, not only do you have to keep the cell body alive, you have to keep this long axon alive. And this long axon can die and cause degeneration. Now, there are many different ways or reasons that axons might change or or degenerate. Some of them are normal, like in during development. Sometimes early on, you'll have, for example, a target tissue that's innervated by two different cells, and during normal development, you want there to be a more targeted matchup between a neuron and its targets. And so one of those will be pruned so that there's more specificity in connections. Other times, it's really disease where the axon can die even without or with cell death. So axon degeneration is very key, and a really interesting process that can cause disease or be normal physiologic. So I don't know how many of you have heard of Cajal. He was one of the great neuroscientists. And part of what he was great at was just looking at the nervous system in using stains that previously hadn't been available. He was actually also the first person to realize that the brain was organized into cells with axons that connect one cell to another. And he realized that there is axon degeneration, so a dying of these axons, even without there being death of the cell. And he tried to look at what this looked like. And these are some of his pictures, his drawings of what he saw as axons were dying. And one of the things you can appreciate is that as you look at this, it is hard to imagine how you quantify it, and which is going to be very important for us to be able to study something and try to prevent it, because we really want to be able to do that in a rigorous fashion. So I've introduced you to the issues about how there can be both degeneration of cells and of axons, and both of those can cause neurologic diseases. I'm now going to talk about one kind of degeneration of axons, which is due to chemotherapy. Now, over the last 50 years or so, our ability to treat cancers has grown enormously. I sometimes people say, oh, we're not very good at treating cancer. This is not, we are not good at it. We're not as good as we need to be. But the ability to treat cancers has been such a difference. For example, when I was in medical school, one of the cancer types that I worked on, medulloblastoma, which is a kid's brain tumor, the survival rate, again, for kids, was about 10%. That same type of tumor, it's now 70%. That means 70% of the kids with it are cured. So that's just one example of many, but we have done a great job in developing and continuing to develop new therapies. But this has come at a considerable cost. So many of the patients treated for cancer exhibit or develop neurologic impairments due to the treatment. These include cognitive impairment, which is sometimes called brain fog, chemo fog, and is seen in about 30% of adults treated with chemotherapy. Hearing loss occurs in patients treated with a particular kind of chemotherapy. And the most common, and the one we're going to talk about today, peripheral neuropathy, which is seen in about 50% of patients treated for cancer. Now, we might have hoped that new treatments, such as targeted molecular agents and immunotherapies, might prevent these side effects, but it turns out that these also have many of these same side effects, particularly peripheral neuropathy. So what is chemo-induced peripheral neuropathy? So what patients experience is numbness, allodynia, which is experiencing something that normally would not be painful as pain. So for example, a light touch like that could be perceived as very painful. That's allodynia. And then just general pain. This occurs in a stocking glove distribution, in other words, primarily affecting the hands and the feet. And this is where the longest axons end, right? We talked about how long those need to be. For some patients, this goes away, but for others, it is a long-lasting effect of chemotherapy, and so really affects quality of life for the cancer survivors. Pathologically, what's seen is damage of the peripheral nerves that go to the hands and feet, for example, with a dying back method of axon degeneration. In other words, the ends of the axons die first. This can occur with a number of different chemotherapies, including the category of taxanes, vinca alkaloids, and platinum agents. And the most common cause of chemo-induced neuropathy is paclitaxel. So what is paclitaxel? It is a microtubule targeting drug. It binds to tubulin, which is the component that makes up microtubules, and it prevents their depolymerization. So it stabilizes and maintains these long microtubules within your cell. And this is one of the major cytoskeletal elements in cells. Now, because microtubules are essential for cell division, and interfering with their function is a very good way to stop tumor growth. So why does this microtubule targeting cancer drug cause peripheral neuropathy? Well, it turns out that because the axons are very long, an important feature is that you need to transport things from the cell body to the nerve endings. And there are motors in the cell. This is kinesin, the other is dynein, that walk along microtubules, which are in the cell, and transport molecules to and from the nerve endings. So you can think that if you target microtubules, you may well interfere with this axonal transport. So this is our time for our first breakout session. I'd like you to go into your breakout room for 5 to 10 minutes. You'll get called back at the end and discuss with the others what components do you think need to be transported in axons, how defects in transport might cause degeneration of axons, and also, and this is always an important feature in science, how would you test these ideas? Okay, and then I'll ask some groups to share with us what they've said on this. So Ishan, maybe send them to their breakout rooms and have them come back in 8 minutes, okay? See you soon.
Yeah, I think everyone is sorted now,
Okay,
hopefully. Um,
great.
yeah, so I
I
think
think,
sorry, go ahead. Did it pop up for you?
hello, I think you forgot to sort me.
It says that everyone was assigned. There should be a button at the bottom. It says breakout rooms, and then if you click it, it should be able to let you join. I think, yeah, I think you're in room two.
Can you find it?
Did you see a button at the bottom? Oh,
She's right.
okay.
There.
Sounds good.
So how was that? Is that about the right level for them?
Yeah, I think that's pretty good. This is actually the first session that I've actually been to as well. I think we're like switching off every week, so there's a different speaker and a different kind of moderator as well. But I think it was great, and I really love the interactivity as well. I think it's good to have all of the students talking and engaging with the content as well.
Uh-huh. I was impressed that they all had their cameras on.
Oh yeah, we have a good bunch of kids.
Uh-huh.
Yeah, they're really engaged, and I think all of the previous sessions have gone really well. They've been asking good questions, and
Uh-huh.
yeah, I think they're super curious, which is amazing to see.
Yeah. Oh, I didn't start to look at the time.
I think we can call them back at 27. I think that would be around 8 minutes.
Okay.
I just need to take attendance for everyone, um, making sure everyone's here. It's been a long time since I've seen Cahal's images,
They're
but
so beautiful.
it's actually incredible how forward minded he was, so far ahead of everyone.
Yeah. Though it's also sometimes you wonder, did he really see everything he drew, or was he drawing what he thought was happening rather than just what he saw? It's a whole other
Mhm.
question.
I mean, I remember many of his diagrams having arrows showing kind of like the flow of information.
Right.
So he had some intuition.
Yes. Which he didn't, I don't know how. So sometimes it was unclear, whether he just was inferring things,
Hmm.
but he was remarkably prescient about so much. It's
Yeah.
really pretty amazing.
I remember, so Cahal used someone else's method of staining, right?
Golgi.
Oh,
Yeah.
Golgi, yes, yes.
And
So
you heard about the Nobel Prize that they both got, that they shared, right?
yes, 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 that's why, yeah.
Yeah.
So Golgi did not understand what he was seeing, really. So Cahal was,
Golgi
if I
developed
remember
the
correctly.
method. He actually had stopped doing stuff. And so what he thought was happening, which is really sort of interesting, is essentially that there was this web of
Hmm.
connection and you plugged into it, sort of like the internet, that you,
Hmm.
and so that each cell was plugging into it and then unplugging. a really
Interesting.
weird idea, but I mean,
Yeah.
for us now, but that was what he thought he was seeing. Now,
Mm.
I think that doing a Golgi stain, which I have done, you realize that it's really hard to tell, like, and you can understand why that misperception would happen. But it's so in some ways, it's more remarkable that Cahal was able to understand what was happening than that Golgi didn't. But
Yeah. It's interesting. I remember seeing Cahal's diagram of the Purkinje cell.
oh,
That was
it's the most
incredible.
beautiful. Yeah. He also, when he was a kid, he wanted to be an artist, but his
Mm.
father said no, he had to go to medical school and be a doctor.
Wow.
So this was his opportunity to use his artistic talents.
Oh, wow. It's honestly incredible. Some people are just so far ahead of their time.
Yeah.
Yeah. I think
But it does emphasize the need for both sort of that artistic, creative portion and really rigorous evaluation. Yeah.
for sure. Yeah. I think we can go ahead and
And great.
close the rooms now,
Okay, great.
if you're ready.
Yep.
Okay, cool. Yes. Will be like 60 seconds.
Mhm. Welcome back.
Okay, I think everyone should be back now.
Okay, great. So was the discussion good? I hope.
Yeah,
Okay.
it was great. We explored a lot of different things.
Okay. So let's talk about um, the first question, which was, what do you think needs to be transported in axons? And Azzy, what did your group come up with?
For all three parts or just the first one?
Just the first.
So for what needs to be transported in axons, we looked it up on Wikipedia, and the main thing mentioned in your presentation were motor proteins, but we also saw that they can transport mitochondria, lipids, and other organelles to a neuron cell body.
Mhm.
So we thought those are pretty necessary for a cell to thrive.
Yeah.
So if there's issues in transportation and you can't get enough lipids or something to a cell, then it's going to malfunction, definitely.
Uh-huh. And mitochondria, because you need energy. Any other thoughts that anyone else wants to add?
I'm wondering if neurotransmitters are also being transported.
Mhm. Yeah, some of them will be synthesized at the axon endings, but some of them do need to be transported. Valeria?
Yeah, these transportation conducts can be overloaded, like because of they can be affected for external factors. We were talking about that in our group. That, for example, if you do not have a good diet, our blood conducts can be, overload because of the lipids. Could that same can affect our brain?
Hmm. Yeah, I don't, there, yes, but it would be more complicated than with the lipids, really. Godfred?
Yeah, we also looked at RNA, where we're trying to say that it is needed for local protein synthesis with the axon, allowing for rapid response to stimuli with repair mechanisms.
Yeah, that's a really good point, and we're going to be coming to that in a minute. Aya?
Also, axons transport electrical signals and neurotransmitters, and signaling molecules crucial for neuronal function and communication.
Well, the transport of electrical signals is not actual physical transport, right? There's a wave of electricity that gets transmitted through the axon that is not actual physical transport. But RNA and proteins and lipids and mitochondria and other organelles are all key. How do you test these ideas? Azzy and Sarah, and then
So
we'll move
for
on. Yeah.
Can I ask my question, or should I
Sure.
answer the?
Yeah.
Okay, so for my question, something we discussed in our group are if like chemicals like chemotherapy causes axon degeneration. What about other things like artificial additives or harmful drugs? Would those also cause this kind of dying back phenomenon, or is it just specifically these kinds of chemicals?
Yeah, no, that's a really good question. It is not just these. Lead is one of the key causes of axon degeneration, which is not in chemotherapy, I'm happy to say. It's just that chemotherapies are one set of toxic agents that we as doctors give to patients, and so we're particularly aware of them. But yes, there are a lot of other factors that are not that can come in the diet, in the environment, that cause degeneration, or cell death as well. Yeah.
Yeah, that makes a lot of sense. Thank you. Another really small, quick question is also, could certain genetic factors make it more likely for someone to be more susceptible to degeneration? Or
Yeah.
does everyone have an equal chance of their brain cells dying if they're exposed to something?
We will come back to that. The answer is that people differ greatly. You know, I said about 50% of patients have this, and in some it goes away, and in others it doesn't. Well, what causes those differences? It's something we're really working on very quickly. I want to get to all the questions, but I also will want to move on. So I see Sarah Ifechukwu and Sarah and Anya, and then we'll call it quits and move on. So Sarah?
Hello. So I was actually gonna ask a question, but to answer the third question, we kind of discussed like fluorescence and kind of determining like with two different groups. So we have one that's treated and one that isn't treated, and so just determining like we talk mostly about neurotransmitters, so determining like how many neurotransmitters actually made it to the end of the axon. Yeah.
Yeah, that's a good way. Yep. Ifechukwu? I'm
Yeah.
sorry, did I say that right?
You pronounce it like almost correctly, but you can just call me Ify. You're very, very close. But my name is Ify, and for the last question, I did have like another question, but I'm just gonna ask that later on. But for the last one, me and my group discussed about how mostly just like talked about like the transporting of different like signals and other proteins. But overall, I did think that like if you were to test these ideas of like degeneration of the axons, you could just like measure the amount of pressure that they feel at a specific point, or if they're even able to like contract their muscles or not. Because like we thought that like, oh, like since these axons are being they're like basically dying, then the cells would like also kind of die in that way, and so like their muscles, which was like an example, would also like not be working as efficiently as possible. So like say like if you got a mice or something and you touched their thigh, if they didn't move their thigh correctly, or it wasn't like they weren't moving at all when you touched them, then there's probably something wrong. And that also like applies to humans. If you like if you had like a pencil to their hand and they felt nothing at all.
Yep. Yeah, and we'll get to that, to how we test that. Yeah. And Sarah?
Hello, Professor. I hope that you are doing well. So I wanted to answer the third question too. For that, I thought about fluorescent markers too. Other than that, the electron microscopy, maybe it can provide some images that can show the abnormalities in the organelle distribution. And also maybe you looking at the electrical activity. And other than that, looking at the cerebrospinal fluid or blood for biomarkers to see the abnormalities.
All really good ideas. Great. And last, Anya.
Hi, so I just wanted to answer the second question about how defects in transport might cause degeneration. And our group found that apparently mutations in genes which encode multiple motor proteins can actually cause neural degeneration.
Yes, and they're very common genetic causes of neurodegeneration. Okay, so I'm going to move forward and we'll come back to some of the questions. This was great. Okay, so how are we want to understand this phenomenon and we want to test it in a place where we can really look at things in a very rigorous fashion. So we use two methods of culturing peripheral sensory neurons to look at these changes in transport and degeneration. What we do is we plate cells, the sensory neurons that are in your dorsal root ganglia, in specific compartments. And then we have them extend their axons. We make them extend their axons, actually, through this divider. Oops, sorry. Which can be 500 or longer microns, to a second compartment where the axon endings are. What this does is it recapitulates the separation between cell bodies and the axon endings that occurs in vivo with the sensory neuron cell bodies in the dorsal root ganglia adjacent to your spine, and then the nerve endings in your skin. And these ones on the bottom, microfluidic chambers, can be used for microscopy, and the top ones can be used for biochemistry. Okay, so we've used that, and we treated either the cell bodies or the axons with paclitaxel. And this lets us say, okay, where is this agent acting? And what we found is that treating the axons with paclitaxel, but not the cell bodies, cause degeneration. And we can quantify this degeneration by staining the cytoskeleton, in this case tubulin, at the nerve endings. And we then take that stain, we binarize the image, which means make it just black and white. And we say, what percentage of the axons that you see here are fragmented? In other words, not continuous, but in discrete fragments. And so that gives us a way of measuring this in a quantitative way so that we can move forward in seeing what helps it without having to have something that completely rescues the degeneration. Now, we thought that things might be moving in the axons, and that might be affected by paclitaxel. And this is looking at one of those things. One of the things that one group said, which is these RNAs together with a protein that helps transport it. So in this case, we have a fluorescent, as you mentioned, RNA binding protein, which is called SFPQ, and it is moving very rapidly in the axons. Okay, so what happens if we interrupt the movements of this RNA and protein? So after we've added paclitaxel to the axons, which we now know causes degeneration, what we find is that the amount of this RNA binding protein in the axons is way low. And instead, it's accumulating in the cell bodies because it can't be transported. Because the RNA binding protein is transporting RNAs from the cell body, from the nucleus, where they're transcribed and generated, to the axons, where they're going to be used to make proteins, what you see is a decrease also in the RNA for that axonal component, which is called BCLW. Because the RNA is then locally translated, you see a decrease in the protein in the axon for this BCLW. Okay. Well, okay, so paclitaxel causes a reduction in transport of these granules and leads to less BCLW, a protein I'll talk about in a moment, in the axons. Does that actually contribute to or cause the neurodegeneration? So here, what we've done again is we're treating the axons with paclitaxel, but before we do that, we introduce additional BCLW protein into the axons. And you can see here that with paclitaxel treatment, you get the degeneration of the axons, and this is prevented by BCLW added back to the axons. So clearly, axonal BCLW is able to prevent this degeneration, suggesting that the loss of transport may have been important in causing the degeneration. Okay, I said I'd tell you what BCLW is. So BCLW is a member of a very large family of proteins called the BCL2 protein family. BCL2 is actually B-cell lymphoma. So it was this protein was originally identified as a protein that is overexpressed and in B-cell lymphomas due to mutations. So BCL2 is was the initial family member identified, and we now know a lot about these family members, and they are critical regulators of cell death and cell life. So BCL2 and BCLW and several other of the family members are pro-survival proteins. They bind to these these pro-death proteins, which and prevent the actions of this. So what does BCL2 do? It prevents the ability of BAX and BAC to make holes in the mitochondrial membrane. In this way, BCL2, BCLW prevent the release of cytochrome C, the assembling of what's called an apoptosome, activation of those cleavage proteins that we talked about at the very beginning, caspases, and so prevent apoptosis. Okay, so BCLW acts just like BCL2 in binding to and preventing the actions of BAX and BAC. Is that what BCLW is doing in our system? And so we tried, instead of introducing BCLW, introducing excess BCL2 or BCLXL into the axons. And as you can see here, and from our quantification, axonal BCLW is the only one that's able to prevent this degeneration. So what's special about it? Well, it turns out that the reason that BCL2, BCLW are able to prevent cell death is through what are called BH, these three BH domains, BH1 through 3, which are shown in gray, which form a hydrophobic pocket that will bind to the effectors and so prevent apoptosis. But these proteins also have this other domain called a BH4 domain, which is shown in green, and these can have non-canonical, not unusual roles that are not just about binding to BAX and BAC. And so we thought, well, maybe the BH4 domain of BCLW is special, does something very distinctive. And maybe that will help us figure out a way of treating this chemo-induced neuropathy. So I worked with my friend and colleague, Lauren Walensky, And one of the things that he does is he generates what are called SABs, or stabilized alpha helices of BCL2 domains. So basically, he takes the protein, the peptide sequence of these domains, and in the middle of it, he introduces these two unnatural amino acids. So he's synthesized this polypeptide chain, just chemically, with these two unnatural amino acids. And then he does this chemical stapling to generate this link between one of these unnatural amino acids and the second one. What this does is it makes the BH domain assume the alpha helical conformation, which is shown here, that it would normally have in the context of the protein. And so we can make this peptide, this very short sequence, act like the BH4 in the protein acts. So I'm going to very briefly summarize a lot of work that was done on this. And I would say that Lauren and I have a paper that was just accepted, and the first author on it was a Harvard undergraduate who worked in the lab. So what we've found is that the BCLW SAB prevents paclitaxel-induced axon degeneration, just like the whole protein did. We figured out how it does that, which is it binds a protein called IP3R, which is in the membrane in the endoplasmic reticulum and regulates calcium moving from the endoplasmic reticulum to the cytoplasm. And this SAB modulates the calcium flux. And this way, we now have a structural blueprint for how to develop a new therapy for CIPN, because we can say, okay, this one worked, this one didn't, and what are the differences? And one of them turns out to be a particular amino acid. So we now can begin to think about how we might treat CIPN by understanding what caused it. So our model of what's happening then is paclitaxel interrupts transport of these RNA granules. Therefore, it reduces the level of BCLW in the axons. BCLW, through its BH4 domain, binds to IP3 receptor, and so regulates calcium in the axon. So what I've told you thus far is really that there's decreased BCLW with paclitaxel treatment. And this is really taking away the brakes that normally would stop axons from degenerating. So is there a gas pedal for axon degeneration? And so to understand whether there's a gas pedal for axon degeneration, we took our cues from what's known about what's called Wallerian degeneration. So in the 1880s, actually, Augustus Waller showed that if you cut an axon, as is shown here, with the scissors, what happens is the distal part of the axon degenerates. Now that makes sense. We talked about how you need transport, and you need to transport things from the cell body to the axon ending. And when you cut it, there's no transport. Okay, that makes perfect sense. But about 10 years ago, two labs, one at WashU and one at UMass, showed that there was a mutation that you could make in cells, and in mice, in flies, in whatever, in cells with a mutation in this protein called SARM1, when you cut the axon, the distal part of it does not degenerate. It does eventually, but instead of this occurring over the course of a day, it will take like a few weeks. And to address one of the things that was said earlier, during that period of time, this is still electrically active. It'll still communicate with muscles, still communicate with the skin. it's working. It's pretty amazing. So SARM1 is needed for axon degeneration. It's the gas pedal. That's in the case of physical injury. What about in the case of chemo-induced neuropathy? And so a colleague at Johns Hopkins showed that mice that normally when you treat mice with paclitaxel, you lose these axon endings in the skin, shown here, and that's what happens. And in mice that lack SARM1, you no longer see that.
Oh, by the way, that's
Yes. Okay. Okay. So SARM1 protects against both chemo-induced neuropathy and Wallerian degeneration. What is this enzyme, this protein that causes degeneration? Well, we're still working on this. So still a work in progress, but here's what we know. It's highly conserved. So worms have it, flies have it, people have it, mice have it. It has these 4 different domains: an armadillo domain, SAM1, SAM2, and a tear domain. So the tear domain is really critical. It's an enzyme that cleaves NAD, a really important metabolite that's involved in energy, and it generates this interesting looking molecule called cyclic ADPR. Okay, that's its function. Before paclitaxel, or before cutting, actually, SARM1 is in an inactive state, which is shown here. And then following injury, it changes to this very different looking active state. So how could paclitaxel be affecting and activating SARM1? So again, we thought maybe it's transport. And so we now made SARM1 fluorescent, and you can see it moving in our axons again. And when we treat with paclitaxel, the speed of movement doesn't change, but the proportion of the SARM1 molecules in the axon that are moving goes way down. So clearly, SARM1 is affecting transport, and paclitaxel is affecting transport of SARM1. Is paclitaxel activating SARM1? So we know that when it's active, it cleaves NAD, it also cleaves NADP, and it produces cyclic ADPR. So when you treat the axons with paclitaxel, here's without and here's with, NAD goes down, so does NADP, and cyclic ADPR goes way up, as you can see here. And these changes are mitigated when we knock down, genetically reduce the levels of SARM1 in these cells. And so now, after paclitaxel, you have sort of the normal levels of NAD, NADP, and cyclic ADPR. Okay, what about the degeneration? So again, if we knock down SARM1, we prevent the paclitaxel-induced degeneration. So here you again have vehicle. Here's paclitaxel. Normally it causes degeneration. In the absence of SARM1, no degeneration. Pretty amazing, isn't it? So what is the role of the SARM1 metabolic changes in causing this degeneration? So before we get into what is the role, let's have another breakout session and talk about why we have this axon destruction enzyme that's present in cells. When might you normally need axon destruction? How is this similar or different from the cell death machinery we talked about? And as always, how can we test some of these ideas? So enjoy your breakout rooms, and we'll come back again in about 7-8 minutes. I'm a little concerned that I'm going slower than I thought.
I think it should be fine. I think the ideal time would be around 1 or like before 1.
Mhm.
But as much as we can get through is good.
Yeah. Yeah. They were very, very interactive. Yeah. I
Yeah.
think the breakout was good, though.
Yeah. Also, if you want, you can join in and hop between rooms.
Yeah. I'm going to just take a little break.
Yeah, that's fair. Also, I meant to ask before if I could record the session.
Yes, they had asked me beforehand, and that's
Okay,
fine.
cool. Thank you.
I never know how much people know, so
Yeah, that's fair. I think
but
they're all at different levels, so
yeah.
kind of being a little bit interactive, but also like making it a little bit simple, but then kind of building up from there is pretty good.
Okay. And I figure it's always nice to highlight the fact that um, an undergraduate was the first author on the study.
Yeah, that's pretty impressive.
Yeah, I mean, she stayed after undergraduate for 2 years to finish it up, and she's now in medical school. But
Wow, nice.
yeah. So you said you're working at Dana-Farber?
Yeah, I'm actually here right now.
Uh-huh. Whose lab are
Yeah,
you in?
I'm in the Wu lab,
Oh,
7th floor. Yeah,
oh,
immunology.
sounds
Yeah.
good.
Yeah, so it's been really fun, super interesting. It's a little bit different though because it's a synthetic biology focused lab. So we kind of do things all over the spectrum. But
Yep,
yeah, it's been super interesting.
good. That's great.
I'm going to grab a quick snack
Okay.
before
Yeah.
the next session. think we can bring them back in around 2 minutes if that's
Okay.
okay with you.
Yep. Sounds good.
Perfect. Okay, I'll go ahead and pull them back now.
Okay. Okay. I hope that was interesting.
Yeah, very informative.
Okay, so what did you think? When do you need axon destruction? Okay, Adiyinka?
So something that my group was like kind of brainstorming was whether there was a neurotropic like virus or something that could have the potential to spread throughout the axon.
Mm-hmm.
Like you were saying that many things can be transported throughout the axon, and I believe that a virus has the potential to be one of them. So
Mm-hmm.
like if we needed to prevent the spread of that virus, I feel like causing axon degeneration could be beneficial.
Yeah, that's a great point. One of my colleagues who's actually who's specializes in working on SARM1, he's actually the person who showed that there are mutations in SARM1 that cause ALS, which makes sense if you think about it. They're hyperactive mutations. That's his theory of why you might need it. Rabies and polio are two examples of viruses that spread rapidly through the axon through transport. And this would be a way of just if SARM is activated by the viruses, a good way of preventing the infection from spreading. On the other hand, C. elegans, the worms, I don't know. So that is a very current theory. So great idea. Other thoughts, Arjan?
I one of the ideas that my group talked about was if the axon gets injured, it would make sense to destroy the whole thing to potentially conserve energy and maybe like
Yeah.
prevent harmful
Again,
substances.
yeah, given that it's 90% of the cell volume, it takes a huge amount of energy. And so if it's not being useful or it's injured, it could give the wrong information, or it could be and it would take a lot of energy. So you might need that. Okay. Emily?
Hi, my group was discussing how, similar to what she said previously, if there's an infection to the axon and it's not functioning properly, but also if you have to replace like the old ones and produce new axons.
Yeah, there is, that's true. It's hard. It's really hard because many of the axon connections form during development. When, say, like in an embryo, the distance between the DRG cell body and the skin is, millimeters instead of the meter in adult. And it's really hard to grow that long. So it could be, but perhaps over shorter distances. Yeah, Melanie? And any ideas on testing these ideas would be great too.
Hi. So for one of the first questions, our group, although this isn't like our answer isn't quite related to like the pathology necessarily, we were talking about how like a lot of I've kind of heard like facts that, oh, we're born with a lot more neurons than as we progress throughout our lifetime and we lose them. So I was thinking maybe axon destruction is kind of it could also be a part of those types of processes where we just refine maybe like our like circuits and like connections in our brain, and we just maybe naturally lose them as we age. That was kind of one of our theories that we came up with.
Yeah, during development, there's both death of cells and loss of axons. So oftentimes you want to refine the connections. So for example, there could be in the visual system, this is really important, where there are columns depending on which eye information comes from. And so you want you're not born with it exactly correctly, but as you see things, this is refined. And so some axons are eliminated, and in other cases, whole cells are eliminated. So yes, during development, that's a really important point. In fact, SARM1 knockouts, animals that lack SARM1, the mice are completely viable and they're okay. The flies are not. It's lethal in flies. And that's because when they move from being a larva to being an actual fly in that pupation, there's a huge rearrangement of the axons. And if you don't have it, the fly dies. So that's a really good point. So Anya?
Hi, so to kind of answer the first question, me and my group talked about how axon destruction can be used for post-injury or disease management, because maybe like promoting axon degeneration can promote the growth of new axons being created.
Yeah, there, particularly in the peripheral nervous system, you can have some regeneration. And if there's a dying nerve, a dying axon in the way, that is a problem. So yeah, that's an important part. So we'll have just a few more and then move forward. So Godfred?
So my group was also focusing on diseases, but it looks like everybody's talking about that. So I'm going to say something different, where I want to talk about in research context. So we can actually take it to study about neurodegeneration. We can also use it to study about functional studies about the neurons. We can actually induce what we call it. What's the term? We can induce axon destruction to actually focus and then study certain functional studies about the brain and the neurons. And then in testing too, I was talking about culture methods. Culture methods as in we can take certain parts of the neurons to the lab, and then we can use neurotoxins and certain other materials to induce it and then see like how new axon degeneration and then cell death machinery can be implemented on the neurons.
Yeah, and how one can use the two together. Now, I know there's still some questions, people with their hands raised, but I'm gonna move forward, and at the end we'll have more time for questions, and we can come back to that. Okay, so. Okay. So one of the questions we had is, we know that SARM1, when it's active, it cleaves NAD to produce CADPR. So is the cyclic ADPR important, or is it just the loss of NAD? So to test this, what we did is we used, actually there are two different ones we used, inhibitors of cyclic ADPR. In this case, it's 8-bromocyclic ADPR. It's very similar, so it's a competitive inhibitor. And what we found, again, paclitaxel causes axon degeneration, and in a dose-dependent fashion, so not at low doses, but at higher doses, 8-bromo-CADPR prevents the degeneration. So it says that an important part of what SARM is doing is not just that it's cleaving NAD, which will lose some of the energy currency of the cell. It's also producing cyclic ADPR. We know a lot about cyclic ADPR from work from Tim Walseth starting in the 1990s. So going back many years, Tim showed that cyclic ADPR in a cell induces this huge calcium spike. It's not the same as the spike induced by IP3, that one that BCLW acts through. And it turns out that part of it is through a second endoplasmic reticulum receptor called ryanodine receptor. And cyclic ADPR will act on the ryanodine receptor to induce calcium to come out from the endoplasmic reticulum to the cytoplasm. So does SARM and cyclic ADPR contribute to paclitaxel-induced calcium dysregulation, which would be what we think is happening? Now, I'm not sure if any of you know about GCAM, which is a genetically encoded calcium sensor. So this is just a very brief introduction to it. So this is genetically encoded. So there's there's been constructed a gene for it. And the protein that is made by this gene in the absence of calcium is not fluorescent, but when calcium binds, and there are 4 sites of calcium binding within this protein. So when calcium binds and you excite it with the correct wavelength, it will then fluoresce. And so we can look and say, okay, what does the calcium in a cell look like? And this shows that, for example, after electrical activity, there's a change in calcium. And you can see here it's going along, and then there's an increase, and that's due to activity. So action potentials will lead to this due to changes in the binding. We can also look at sort of the baseline levels of calcium using this genetically encoded calcium sensor. And so here we put in a genetically encoded calcium sensor, and we added something so it would go directly to the axon rather than staying in the cell body. And we also put in another protein so we could look at the same time as we were looking at the calcium, look at the axons overall. You can see here that exciting it at the correct wavelength, in response at baseline, there's very little very little visible calcium because of the concentration it's at. And following paclitaxel, this increases at the same time as the amount of axons, which you can see in red, are decreasing. And this compares to control. At control and at 48 hours, we're not seeing much change in the baseline calcium, and you see actually growth in the axons. So with paclitaxel, you get this increase in calcium in the axon cytoplasm. And again, knocking down SARM1 prevents this increase. So you see here the quantification of the calcium levels, and you see here what happens with SARM knockdown and paclitaxel. What about cyclic ADPR? Again, using that antagonist, what we see is paclitaxel causes this change in basal level of calcium, and this is partially blocked by 8-bromo-CADPR. Okay, so to sort of summarize what we found in these studies, looking in culture, is the following. We know that paclitaxel affects transport. It affects transport of RNA granules, such as SFPQ and BCLW. And normally, BCLW in axons is made, and it binds to IP3 receptors through the BH4 domain and regulates calcium. Paclitaxel also affects the transport of SARM1, and paclitaxel activates this enzyme to cleave NAD and produce cyclic ADPR, and so affect ryanodine receptors and again change calcium levels. And again, summarizing what I'm not showing here, we know that calcium will then bind to these proteases called calpains that are in the axoplasm, in the axon, and that activation of these proteases will cleave proteins in the axon, particularly the cytoskeleton, and cause degeneration. Now, what's cool here is there are a number of places we now can think about interrupting this. We can think about using our knowledge of that BH4 domain to try to replace the endogenous BCLW with a chemical or another BH4 mimetic. We can use an inhibitor of this SARM enzyme or an antagonist of cyclic ADPR, or we could consider using calpain inhibitors. So understanding the mechanism really gives us insight into what we can do to treat a disease. So I'm just, again, this is going to be the last breakout session, and just to talk about calcium, because again, one of my colleagues says, it basically in neuroscience, calcium is everything. Calcium is critical in so many ways. So as we're looking, where within this cell do we want to measure calcium levels? Is that in the axon, in the endoplasmic reticulum, in the mitochondria, in the nucleus? What are some of the functions that calcium regulates? And does the source of calcium matter? Does it matter whether it came in through one kind of channel or another? Does it matter whether it came from outside the cell or from stores inside the cell? What do you think? So again, last breakout session. So enjoy your discussion, and we'll come back in like 5-6 minutes. Bye. Good. So what other things do you do with these high school students over the summer?
So basically, everyone is kind of paired up one-on-one with a mentor, and they're kind of working on their own, like literature review on a topic of interest that's shared between their mentor and themselves. And then we have like these speaker sessions, and then we also have like workshops where we talk about various aspects of research, like, let me sort this person, like really asking the right questions or experimental design, scientific communication, like key skills. Like it's really supposed to be a foray into research for high schoolers to really like get them excited, get them a little bit more aware of like what research actually is and kind of like the excitement about like really solving a new problem and being like the first person to know something that is just like it's pretty cool.
Yeah.
At least in my opinion.
Oh, definitely. Yeah.
Yeah. I remember my first research project. It was so fascinating. So I used to work in a comp bio lab in high school, and it was just so fascinating, like seeing all of the results at the end and like really just feeling like you're the only one who knows this information. It's like it's pretty cool.
Yeah, that it is incredibly cool.
So which department in Dana-Farber are you in?
In cancer biology.
Ah, okay. Is that fourth one? Or
We're located over in the Longwood Center.
okay. Got it, yes.
Well,
Okay.
mostly. Part of cancer biology is still on Smith 9
Okay.
and 8.
I see, yeah.
But most of it is in Longwood Center now. Yeah.
Nice. Got it. Yeah, it's Dana-Farber is it's actually a lot bigger than I expected. There are a lot of different labs, different groups. It's really cool.
And compared to other cancer centers, one of the really exciting things to me about it is that it tries to, and I don't know that, but it has, have an equal emphasis on research and on clinical care.
Yeah, for sure.
UT Southwestern and Sloan Kettering certainly have larger clinical care groups and networks, but Dana-Farber, given the size, really emphasizes the idea that it's about the research as well, and really they work very closely together.
Yeah, I think that's super important. I was at Lurie's in Chicago, and it was super beneficial to like be able to interact with the patients that you're working research for, and also it was just super smooth. If like I needed a specific sample, I could just ask one of the surgeons across the street, and it would be super easy. But it's so useful.
Yeah, and so important. Well, different people have different motivations, and I think sort of understanding the basic biology is really key. And I don't mean to dissuade that. I've always been very motivated by the idea that that the research not only is interesting and novel, and you're learning things, but also will have practical repercussions for health. So, for that motivation, yeah.
Yeah, for sure. I think for that reason, I'm actually really interested in becoming a physician scientist, MD, PhD route. It's a long journey, but I think it's definitely rewarding.
Yeah. After Sophia, the student who I mentioned, who was the first author, the next undergraduate in my lab, Ezechuku, just started at Vanderbilt in the MD PhD program.
Nice. Very cool.
Yeah. So.
Should we start letting everyone back in?
Yeah.
Okay, sounds good.
Welcome back. Okay. I'm going to start calling on people who have their hands raised and have not spoken before. So Shuria, what did your group think?
Yeah, so within our boundaries of the first question for now, we basically said that measuring calcium can be in multiple parts of the cell. So in the nucleus, in the mitochondria, I specifically said in the mitochondria, if there's an excess amount of calcium, then it could lead to cell death. So yeah, that's basically what we said there. And also in the ER, but yeah.
And one really interesting thing about the ER is, you think about an axon, which can be like really small. It's like a micron in diameter, could be that small. And there's an endoplasmic reticulum that goes all the way out to the end. It's pretty amazing to think of. So that essentially it's not really a circle, it's sort of a donut shape in the axon. But all those places are really critical, and they're not always the same within a cell. So, Catherine?
Oh, wait, yeah. So like, I actually have a couple questions. So like, does like an insufficient blood level of calcium lead to like endoplasmic stress? Like,
Yeah. So there's very tight regulation of the calcium levels within the cell and within different parts of the cell. So yes, there can be a range of calcium levels in the blood, and how those are regulated is a whole other story. But that doesn't it doesn't immediately change the cellular level of calcium. So there's a lot of range where you can really equilibrate. So while the regulation of blood calcium and the regulation of intracellular and ER and mitochondria and nuclear calcium are all sort of distinct. Obviously, they all impinge on one another because it's hard if there's, for example, if there were no calcium, you can't, in the blood, you couldn't get calcium into the cell. Or if it's really high, it's hard to keep it out. So that gets into the question of, does the source of calcium matter? So whether it comes from the blood, or whether it comes from the ER to the cytoplasm, or whether it comes from the mitochondria to the cytoplasm, or whether and what kind of channel it comes in through, all of those things turn out to be really, really critical. Augustus?
Yeah, I think that was kind of my question too, but I was just wondering if like too much calcium, like what kind of problems that can pose, and like if hypercalcemia and like that can play a role in this.
Yeah, so again, we're looking at things that are happening not across every part of the body, but in the neuron. So there's not, in these contexts, even though we saw the changes in calcium in the axons, there's not change in the blood. Hypercalcemia, however, causes major problems. It's usually an endocrine problem due to parathyroid hormone, or else due to kidney disease, because you're not able to get rid of calcium. And those do cause major problems, but not specifically axon degeneration. Hassan?
Yeah, I had an answer that I think like answers the first two questions. So I think like one place that we might need to measure calcium levels is within the nucleus of specifically nerve cells, because I know that creation of a calcium concentration leads to an action potential signal, which does lead to neural transportation and neural communication. And so I guess that kind of answers like the first two questions, like what are some functions that calcium also regulates?
Yeah, so calcium is critical for neurotransmitter release. It's also critical for propagating and it's also critical for initiating the electrical changes that get the action potential started. So it's critical at both those sides. It's also critical in the nucleus, as you said, for regulating gene expression. And so there are changes in gene expression that occur in response to activity that require calcium. Okay, and Anya?
So I found an answer for the second question. So the top 3 functions I've found which calcium can regulate are coagulation, muscle contraction, and bone health.
Yeah, and none of those are within the neurons. So although those are very key elsewhere, not though it just points to the fact that calcium in different cell types and different organ systems has such critical and such different functions. So Ariane, and then Ozzy, and then we'll move on.
Kind of like what I kind of like guessed was that since calcium is like a good conductor of electricity, it could help in like passing in the charges between different axons and like act as like a messenger.
Mm-hmm. Yeah, it is really key for, again, as I was just saying, for initiating the action potential, which is primarily due to regulation of sodium and potassium levels, both extra and intracellularly. And also for then translating that action potential into neurotransmitter release so that one cell can send a signal to the next. So yes, very, very critical. And that's why one of my friends says calcium is always the answer. So Ozzy?
So something my group discussed that I didn't learn before is that I heard if there's too much calcium buildup in the mitochondria, it might lead to cell death or something. And I think that is an important place to measure besides the endoplasmic reticulum because of how the buildup can be detrimental. And also, for the last question of how the source of calcium matters, I heard Harvard Health published a blog that said calcium citrate and calcium carbonate are absorbed by the body differently, and those can be found in different supplements and foods, which implies that certain calcium-rich foods and supplements are better than other ones. So I don't think it's like feasible to just eat a block of calcium and then you get the same health benefits as drinking milk, right?
Yep, that is very true. And just to talk about where within the cell, we can take, because it's genetically encoded, just like we took the GCAMP and we targeted it to the axon, in our case, the axon membrane, so it would record what's in the cytoplasm in the axon. We can also target it to the endoplasmic reticulum, to the nucleus, to the mitochondria, actually to the peroxisome too. So there are a lot of, to the synapse, so lots of different places. And so you can measure it at distinct locations because of these wonderful genetically encoded calcium monitors. Okay.
That's really interesting. Thank you.
Okay, now. Okay, so we've been talking about using our understanding of the biology of the system in these very reductionist systems, individual cells and cells grown in these culture systems. And how are we going to then translate it to give us ways of treating people who have this problem of chemo-induced neuropathy? So we have moved to mice where we can test things. So in our case, what we have is we have a mouse with breast cancer, and we have it with a specific kind of breast cancer, which is sensitive to paclitaxel. So what we did is we have the mouse, we treat with paclitaxel on a very specific schedule, which is basically twice a week for 3 weeks. And we also give inhibitors of SARM1 or of cyclic ADPR, and again, on a very specific signature. And now we can start to see if what we now understand about the process can give us new ways of treating this chemo-induced neuropathy. What we can do is we can analyze in our mice the behavior of CIPN, specifically allodynia, the feeling that something that's normally just light touch becomes painful. We can analyze skin innervation and actually look at axon degeneration in the skin because we know that's where the skin endings are. We can also analyze tumor size as well as proliferation and cell death within the tumor to make sure that any intervention we're doing is not interfering with the treatment, because we know very well how to prevent chemo-induced neuropathy, which is not to give chemotherapy, but then the patient's going to die of their cancer, which is not a good outcome. Okay. So this is an illustration of how we measure allodynia for mechanical pain. So we have a little machine that is going to be poking the mouse's foot with a very soft fiber. And first it'll just do a very soft poke. We can measure the force that we're using to poke it. And we can see, is it causing distress to the animal? Does the animal just not care about it? Do they take it up and go the way you do when something hurts? Do they withdraw it? So we can measure whether there's allodynia, whether what normally would not be causing pain is causing pain. So here's what we did. We did that in our mice. And here's before and after injecting, instead of paclitaxel, we just injected saline. So just a vehicle. And you see that before and after the injections, there's no real difference. You need a specific amount of force before the animal will withdraw its paw and shake it. After paclitaxel, what you see is the animal really responds to just a tiny touch and it goes, ow, that hurts, which is what happens with our patients, where just like literally their feel of their clothes can feel painful. So that's what happens after paclitaxel. Now, the animals that were treated with 8-bromo-CADPR, the antagonist for CADPR, it prevented that change in behavior. Similarly, the SARM inhibitor really reduces this behavior of pain. So that's cool. What about the skin innervation? And again, here's the skin innervation visualized by staining the nerve endings. You can see it in different parts of the skin. You can see that after paclitaxel, we lose a lot of the axon endings. The animals that were treated with CADPR, we have much less loss of skin innervation, which is shown here. Similarly, the SARM inhibitors really largely prevent this loss. So they are doing what they did in vitro, which is great in terms of thinking of these as potential therapies. But again, what about the tumor? Okay, we can also, because we're doing this in a tumor that's susceptible to paclitaxel, what we can do is measure the tumor over time. You can see here that the tumor is growing. Treatment with paclitaxel in this blue line really reduces the growth. It doesn't completely eliminate it, but both times you can see how effective this is, which is why it's so often used as a treatment. If we add in the CADPR inhibitor, what we find is it doesn't affect the normal growth of the tumor, and we still are able to have the efficacy of paclitaxel. And similarly with the SARM inhibitor. I want to just note one thing that we're looking at right now, which is most of the way this is completely the same. And for both the CADPR inhibitor and the SARM inhibitor at the very end, They see it's not statistically significant, so I don't know whether it's random, but we saw it twice. So I'm a little concerned that there may be some divergence here. And so it tells us that we need to now do the same experiment, but go longer to see if after 4 weeks, it's really preventing the efficacy. But we're still in a mouse, and we want to think about our patients, not just the mice. So a question that someone asked early on is what makes some patients more vulnerable than others? And there's some things we know. If you have a larger dose of chemotherapy, that causes more neuropathy. That's not so hard to imagine. How long you give it for, whether you give just one chemotherapy like paclitaxel or a combination like paclitaxel and carboplatin, two potential neurotoxic substances. Age, older patients are more vulnerable. How active the person is, whether this is direct or indirect, we don't know, but actually their BMI, how their metabolic state and obesity do correlate with patient vulnerability. And then there's this clear evidence that genetics plays a role, though we don't really understand it. So one of the things that we're currently doing, and this is very much work in progress, is we've collaborated with one of my clinical colleagues, Joyce Liu, who oversees the ovarian cancer patients. And what we did is we took 10 patients who were treated with Taxol and carboplatin and developed such severe symptoms of neuropathy that they had to actually stop therapy. We don't like to stop therapy because it makes it less likely that you're going to be cured, but sometimes the toxicity is sufficiently bad that we do that. So those are the first 10 patients. She also identified 10 patients who were matched for age, race, and approximately for BMI, who had very mild symptoms or no symptoms. And so we now have 10 pairs of patients with severe CIPN and control. And what's important, both of these were challenged with the same regimen. From these 20 patients, we've generated iPSCs, which is induced pluripotent stem cells. And we can then take those stem cells and use them to generate sensory neurons and test the difference between our patients. So how do we induce pluripotent stem cells? This is a very quick summary of what was a Nobel Prize winning discovery by Yamanaka. Basically, you can take adult fibroblast or adult blood cells. You introduce particular cocktails of genes that reprogram them so that they are no longer this differentiated kind of cell, but are a pluripotent stem cell, more like an embryo. We can then take these iPS cells, and make blood cells, pancreatic cells, nerve cells of different kinds, fat cells, muscle cells. It's really amazing. So this is great because now we can take just the blood from a patient and use that to make neurons that have the exact same genetics as that patient. This is how we differentiate the iPSC cells into the sensory neurons. It takes time and a whole host of different culture conditions, but over time, we get them to be like neurons. And we can look at them over time and show that they slowly develop, reprogram them going through the process of becoming neuronal-like, starting from the iPSCs, becoming ectoderm, becoming neuronal-like, becoming the neural crest, and then going from the neural crest to sensory neurons. And we can monitor this progression over time by looking at markers for these different cell types. It's really amazing that this is possible. I will say it's not as good as it sounds, because the sensory neurons we make aren't completely the same as normal sensory neurons, but they're pretty close. So again, work in progress, but we've begun to test these. And so again, we can do exactly what we did with our mouse cells in culture. We can take the patient or the control and treat them with paclitaxel, treat them with carboplatin, or the combination. And you can see here, again, looking at the axons, that paclitaxel causes degeneration, so does carboplatin, and the combination is even worse. But when we quantify it, and again, this is why we so much wanted a quantifiable assay, what we see is that comparing the patient and with severe CIPN and the control, they respond similarly to paclitaxel. They respond similarly to carboplatin, but this combination, we see this huge difference. We don't yet know why, but it's really exciting for us that we're beginning to tease apart some of those genetic causes of variability. Okay, so I've told you about degeneration in general, about how paclitaxel interrupts transport. It includes transport of RNA granules and also of SARM1. There are multiple things that probably contribute to paclitaxel-induced degeneration and changes in calcium. And as we use our reductionist thinking and the biology of the system, we can then begin to translate that into ways of treating patients and preventing this degeneration, either using BCLW mimetics, SARM inhibitors, or cyclic ADPR inhibitors. And importantly, it seems as if we can treat the degeneration without preventing the treatment of the cancer. And we can also study this in human cells so we can understand the variability between patients. So this is my lab and different people and funding sources and collaborators. Because one important thing about science is each person is doing stuff on their own, but we're really working together, and a lab can't do everything. And so having collaborators is really key. So with that, other questions that you have? We have about 10 minutes, so I can continue to answer questions. So thank you all for listening. Okay, again, I'm going to start with people who haven't talked before. So Joshita?
So wondering if paclitaxel-induced neuropathy makes patients more susceptible to neurodegenerative diseases like Alzheimer's and Parkinson.
Really good question. And the answer is we don't know. So there has been a suggestion, and I believe some groups are working on this currently, that the other major neurotoxicity, the cognitive neurotoxicity, is really associated with later Alzheimer's, but we don't know because it hasn't really been looked at in depth for either the neuropathy or the cognitive acute functions. This is something that initially, as we developed ways of treating patients, everyone was so excited that the downsides weren't really appreciated, and we're really just getting to those now. Yeah. So Catherine?
Yeah. I actually have two questions. So like the first one is like based on your slide about like what makes patients vulnerable to neuropathy. So like, are the treatments same given to both like the juvenile and like adult groups?
Ah, really good question about differences between juvenile and adult groups. So, the cancers that kids have and those that adults have are very different. And so some of the chemotherapies are the same and some are different. So, for example, paclitaxel is rarely given to kids. Vincristine and vinblastines, which also act on microtubules, are given to kids. So that's a huge difference. The percentage, again, age is a critical aspect of vulnerability, and kids are less vulnerable than adults, but not completely resistant. Did that answer your question?
Yeah, that makes sense. And my second question is kind of unrelated, but like, what classes, no, like, are there any classes that I can take in college that can help me like become so successful in med school?
Oh gosh. Um, yeah, you should major in what interests you, take the classes that are required for your major. You'll get to know stuff about whatever it is, and you'll become, and yeah, it's a lifelong learning. So you're not going to learn everything in college or in graduate school or in medical school or ever. So yeah, just
Oh,
do
that
your
makes
best.
sense. Okay, thank you so much though.
Sahaj?
Yeah, hello. Your presentation was really good, by the way. My question was, what role does like oxidative stress play in the neurotoxicity? Like going back to Joshita's question regarding the oxidative stress as well, and like Alzheimer's, like what exactly does oxidative stress play on neurotoxicity?
Yeah, the answer is we don't know. There is oxidative stress that you can measure in the cells. Is that causative? Is that a consequence? Where does that stand relative to BCLW? Where does it stand relative to SARM? Where does it stand relative to CADPR? Yeah, we just don't know. It does occur, but its role isn't clear. Yeah. There are a lot of, the other thing to say is there are a lot of ways that a cell can be screwed up because it's such a complex machinery. I mean, there's so many different ways it can be screwed up. And once you screw up one thing, everything starts to go wrong. And so what's really hard is figuring out what starts it and where you can intervene. And those are the places we try to really focus. Yeah. So Anya Krislov.
Hi. So I was wondering, this is more of a broad question, but is there any world where induced axon degeneration can replace anesthesia, and like how does that compare? Would there be any
Ooh.
benefits to that?
Yeah, no, you wouldn't want to do that.
Mhm.
Because if you cause axon degeneration, then to recover from that, you have to repair it. And that's really, really hard and really complicated. And so, yes, so the numbness that you feel is also not complete absence of sensation. So it's paradoxical that patients with chemo-induced neuropathy have both numbness and pain. Like you say, well, that doesn't make any sense because they're numb. How do they feel pain? But what's true is that the axons aren't working. The axons that are there are not working properly. So even though some of them have died, and there's degeneration, there is also abnormalities in the axons that remain. And so that would be a terrible idea and wouldn't help for preventing pain. There are a lot of great ideas that are really novel about preventing pain. And yeah, it's something that's a fascinating field. But yeah, axon degeneration is like, I don't know, shooting yourself because you feel pain. Yeah, you won't feel any pain, but you're dead. That's not a great solution.
Okay, thank you.
Sorry for the dire comparison. Andrea?
Hi, how are you? So I just had a quick question about public health, and it's a career that I kind of want to pursue in the future. So I was just wondering whether you had any experience that you could talk about related to like public health and cancer research, like how you would like incorporate the two if you have.
Yeah, that's a huge area. So epidemiology and cancer is obviously very active field, trying to understand at the global level what causes cancer rather than at the individual level. There's also a whole field of quote outcomes research, which in some ways what I'm talking about informs, which is okay, if you think about the outcomes of a particular treatment, whether it's, for example, the new CAR T cells, and you think about how much they cost and how difficult it is to do them. How do you think about the cost benefit of that? Those are areas that people, there are many MDs who have also have a Master of Public Health and are very involved in that. That has not been something I've ever really has been part of what I do, but it's a really important area. Adeyinka?
Hi, my question is like more so about like how does the specific interaction between like what you were talking about, like the SARM1 and the CADPR specifically contribute to like the elevation of like calcium levels and
Yeah, how does these different things, how do they actually cause it? Yeah, really hard question. We can say that SARM and CADPR, active SARM and CADPR do contribute to the changes in calcium. We do know that if you eliminate ryanodine receptors, which are one of the targets of CADPR, that effect is much less. But really working out the intricate mechanisms is something that we're still working on and will be for a while.
And this is kind of slightly like not off topic, but I feel like every single time that I hear of experiments being done or like studies being done, it's always specifically on mice or with mice. So, although this is not like particularly on topic, can you explain why it's usually mice and not another animal?
Yeah, really good question. And that is really important. Okay, so what are the advantages of mice? There are a lot of other model systems people use. I mentioned flies, I mentioned C. elegans, worms, nematodes. Some people use primates. It used to be that dogs and cats were used. Okay, what's the advantage? And rats are also used. So what's the advantage of mice? First of all, they're little. That's really a big advantage. They don't take up so much room. They're very costly for researchers, but we can keep them in cages and they're reasonably happy and healthy there. And we can have large numbers of cages. So that's number one. They're small. They don't take up so much room. You can have a lot of them. Second thing, mice in general live in very small colonies, so they can be inbred. In other words, you can literally breed brother to sister for 20 generations and get viable mice that don't have all these weird homozygous mutations that cause problems. What that means is we can have and we do have genetically defined populations of mice that all have very, very, very similar genetics. Unlike humans, where we're much more outbred and where we're much more diverse. So you can have mice that are inbred. Secondly, so that was secondly. Thirdly, we can manipulate the mouse genome pretty easily. Well, what's now pretty easily. We can make what are called knockout mice. We can make mice where we introduce a mutation in a gene so it's gone. We can modify a gene so that it's changed, so we can replicate a human disease in mice, again, pretty easily. So that's another real advantage. Fourth advantage is that that we can, they develop fairly rapidly. So gestation is 3 weeks. And after birth, they're sexually mature at 6 to 8 weeks. So it doesn't take forever to do our experiments. Let's see. You can teach them. So for neurologic questions, you can look at, did they learn how to go through a maze? Did they learn this? Did they learn how to stay on a precarious ladder? So there's a lot you can test in them. They're mammals, unlike Drosophila and nematodes, so they've got a lot of similarities to people. And finally, they've been used for studying for so many years that we have a lot of specific mutations in them. I said, after SARM1 was identified, it turned out we had a mutation in SARM in mice made and available to everybody within 4 years. And finally, there's a place called Jackson Lab that supplies mice, and they're not that expensive to buy. And they have so many different mice so that they can be shared throughout the community. Those are all the good reasons to use mice.
Okay, thank you.
Sure. Ariane?
Yeah, so I was just wondering, like, are there any like recent studies dealing with like stem cells, like mesenchymal stem cells with like cancer treatment?
Oh God, yes. A ton. There's so many stem cell and cancer treatment things. Yes. I again, yeah, it's a hugely active field. Arjan?
Thank you for the presentation. My question is also a bit off topic. The iPSC cells you mentioned, they could turn into any type of cell. So why aren't they used for blood transplants or organ transplants?
We're hoping that they can be, but it's not just about having, so for blood transplants, they're beginning to be clinical trials using such cells. I didn't go through this, but to make the iPSCs, we actually use a bunch of oncogenes. There's concern that some of these would turn into cancers in people if we kept them going we keep them going for months in the lab, but not years. And then the other thing is you have to not just make individual cells, but assemble an organ, which is very complicated. So yes, it's something that people are trying to do, are really interested in doing. We don't have it together yet in a way that we know is safe and effective. But it's an area very active, very, very active research at both at the basic science level and at the clinical level. Yeah. So Anya?
Hi, so my question kind of revolves around what I'm writing my research paper about for this program. So my question is, are there any current therapeutic strategies for managing CIPN symptoms? And if so, what are they? And like, how do they work?
Yeah. There were, gosh, a while ago, there were 14 different clinical trials, and 13 of them showed no efficacy. One, duloxetine, which is an SNRI, it's an antidepressant that's serotonin and norepinephrine reuptake inhibitor, had some efficacy, but really pretty minimal. So that is the one thing that's been approved for treatment. What are the things that are in trials? People are trying things like acupuncture, yoga. I told you that BMI and physical activity accorded with the level of susceptibility. So people are trying exercise programs. SARM1 inhibitors, I believe they just started a clinical trial with some of those. Those are the things that they're trying. Oh, and then some diet manipulations. Yeah. And Godfred?
Your lecture has been very, very educative, and for me, it has really learned a lot. So my question is on the calcium we did in the breakout session. I want to ask that, you see, they were calcium at different places of the body. And so I want to, and then when we asked of if their sources matter, it was like, yes, their sources really matter. So I want to see that their sourcing being matching, is it that there are different levels of calcium at different sides of the bodies, or is the same calcium at the different parts of the body?
Yeah, really good question. So within the body, there are places where there are different levels of calcium. Obviously, bones are one where it's really where there's much more calcium than, say, any other part of the body. So it's different parts of the body and also different parts of an individual cell, right? So the level of calcium in the nucleus, cytoplasm, ER, and mitochondria are all different. So the mitochondria and endoplasmic reticulum have higher levels of calcium usually than the cytoplasm, and the nucleus, I'm not sure, but again, it's all of those can be distinct within a single cell. And then in the other thing is like when a neuron is active, electrically active, and is releasing neurotransmitter, there's a huge increase in calcium. So time, place, what it's doing, all of those are important. Yeah. And Sarah, and then that'll be the last of the questions, I think.
I also put this question in the chat, but I had a question just about the alpha helices.
Oh.
So how did you know which amino acids to put in to get that function?
Yeah. So we just took our cue from nature. We started with BCLW because we know that worked. And so we just incorporated those, and then those unnatural amino acids that I mentioned. So Lauren put them, I didn't do this. Lauren put them, they have to be, because of an alpha helix, they have to be either 4 or 7 amino acids apart, just because of the way an alpha helix coils. And so he did he took that sequence and then replaced the amino acids with the unnatural ones all the way through, found out which one works best. Then after that, we took that SAB and we said, okay, we're going to change every single amino acid to alanine and see which ones are most important. And so we could figure that out. yeah, so it's a long process.
Yeah.
Okay. And I see that Krishna sent me a message. So yes, I will do that. And otherwise, thank you all for your great questions and for your involvement. And, you know, I was saying to Ishan during one of the breakout sessions that, you know, science is really interesting, and figuring out what motivates you is really important. For me, I find the biology super interesting, but I also want things to have the health and clinical implications. And so for me, those two things need to be there. And that's what I try to do. So good luck with the rest of the summer, and good luck with moving forward, and enjoy science. So
Thank you so much,
yeah.
Dr.
Thank
Segal.
you so much.
Thank you.
Thank you.
Thank you so much.
Bye.
Have a good day.
Thank you.